Hi everyone. Today I'll be speaking about bacterial strains in biotechnology regarding a drug that was developed with sulfonation of anilines. So, in the 1930s, two chemists by the name of Fritz Michi and Joseph Clarer were working on a research lab in a dye company called IG Farben Industries. And they were looked into some dyes that were being used and discovered that it had medicinal properties. So azo dyes are organic compounds with the aryl and substituted aryl groups, and they are used for textile and many 60 to 70% dyes in foods and textile, leather. A problem was that some forms were carcinogens, like benzodiazepines. Pronicil was discovered in 1932 by a team at the Bayer Laboratories. Also, they have produced medications such as aspirin. Um, it's a drug that converts the active metabolite in the gut by the gut. And it attacks the broad spectrum of the gram-positive bacteria. So another definition of a prodrug is a chemically modified inert form of the drug, which means it's inactive before bioconversion through metabolism that alters the prodrug's molecular structure, producing the active form of the drug. In this case, it is the Prontosil that is the inert form, and through metabolism alters the structure and changes it into the active antibiotic form. So Prontosil is a red chemical dye with antibiotic properties, but it does not work without metabolism. Metabolism alters the molecular structure of Prontosil, the active form, and changes it to the active form sulfonilamide. This powder was used a lot during war. It was sprinkled onto the wounds to prevent infection. So Dogmack discovered that azo dyes inhibited the growth of bacteria in mice cells, but outside of the mouse, it did not affect the bacteria. This is considered a prodrug because the chemical enzymes activate it within the body. This success enabled curing disease such as streptococcus, which is the main cure for the president's son, Franklin D. Roosevelt. And the drug paved the way for current chemo medications today. So hey guys, welcome back to the PowerPoints. I did very much enjoy the video. Uh, History is not my favorite subject, but I found that very interesting about the Prontosil being a cure to help Franklin D. Roosevelt's son. Okay, so now we're going to talk about how prodrugs help create new drugs. They improve the membrane permeability, absorption, and distribution. They alter metabolism and the side effects and toxicity of any drugs. One prodrug, another example is the inactive form methamphetamine, and it was used to treat bladder and kidney infections. So in order to break that down, actually was the acidification of urine. So the acidified form becomes formaldehyde, the active form of it. In conclusion, the discovery of prodrugs through azodyes led to the discovery of Prontosil, an antibiotic which helped cure such diseases as streptococci, which was a cure for Franklin D. Roosevelt's son in the early 50s, and prodrugs are the inactive form of the drug until they are biodegraded through metabolic enzymes. They're also very effective for developing new drugs today, such as anti-cancer meds and antiviral, because they help improve the permeability of membrane, the solubility of the drug, and the toxicity and side effects of the drug. Thank you. What I found interesting about prodrugs was that they work with enzymes in vivo in the body and not outside, not in the test tubes. It has to be in a living form